was thinking, okay, so this means that ADP means that there's low amount of energy, and this means that we would want to go into the citric acid cycle, which means we want to make energy. We're low on energy, but we're efficient or sufficient in glucose. And this calcium is because muscle is contracting, and it's pretty much saying, okay, we need more energy because I'm working out and I'm repelling or suspending, not repelling, letting go a lot of calciums to allow the citric acid cycle to happen so more energy can be made. Does that make sense so far? Mm-hmm. Okay. And TCA is used in porphyrin synthesis, gluconeogenesis, amino acid anabolism and catabolism, and fatty acid buildup and breakdown. And if you have buildup and breakdown in the same system or cycle, then it's called amphibolic. And it's also involved in ketogenesis as well as oxidation in the electron transport chain. So this cycle is super important in regulating a lot of things in our body, especially our carbohydrates. Any questions so far?
You're gonna get to the phosphatase?
Yes.
Okay.
Well, the oh yeah.
That's another one, right?
Yeah.
Okay.
You're right. So, like I said, that was 4. So this is 1, 2, 3 is the kinase, mm-hmm. and the phosphatase is actually pretty simple. It's if you have insulin and calcium, that will activate the PDH phosphatase, and then that will take away a phosphate group from the PDH and activate it.
So there's 2 mechanisms to regulate PDH kinase, basically?
Yeah.
Okay.
In terms of inactivation, activation, and then okay. I don't think there's anything to deactivate the PDH phosphatase.
Yeah. Okay.
Like glucagon does not deregulate or deactivate.
Okay.
So yeah, I think the main bulk of this lecture was regulation, especially that regulation of the pyruvate dehydrogenase. Any questions so far? I hope you're not like too focused on the video. Okay, so I'm gonna erase this part just to make room. So another issue that came about was this oxaloacetate. How is it replenished? And that is something called the aponeurotic apoplerotic reactions. So since we're using oxaloacetate, we have to replenish them to recycle it to make this cycle happen, and that is called the apoplerotic reaction. So there's 2 apoplerotic reactions in the citric acid cycle, and one of them, generally speaking, is the 4 to 5 carbon 4 to 5 carbon replenished amino acid degradation. So it's just saying that within these things there's 4 or 5 carbons, and they're going to go into becoming amino acids, and they're also going to go in from amino acids. And that's pretty much what it's saying. That's how it gets replenished. As well as the main thing is oxaloacetate. How it gets regenerated is the pyruvate carboxylase. Like I said, we have the PEP from glycolysis that turns into the pyruvate, which if it decides to go into the citric acid cycle, it will become acetyl-CoA. But if it doesn't, it will go back into PEP, and one of the ways it does that is through pyruvate carboxylase. And if it doesn't want to do like the gluconeogenesis, this oxaloacetate can go back into the citric acid cycle and be recycled back. Remember, carboxylase is a very interesting enzyme. It's not pyruvate dehydrogenase. Two very distinct. Pyruvate dehydrogenase, 5 subunits. Carboxylate has just one reaction, pyruvate to oxaloacetate. This oxaloacetate has 2 options to go to. It can be gluconeogenic, creating PEP, or it can go back into the citric acid cycle, replenishing the oxaloacetate that was used in the citric acid cycle. And TCA cycle generally is going to be stage 3, even though it was part of stage 2. And a good thing to note is since it happens in the mitochondrial matrix, red blood cells do not undergo TCA because it doesn't have mitochondria. And then there are some inhibitors of this. So I'm gonna go on a chair and tell you guys this. So some things that not only is pyruvate dehydrogenase really regulated via not having those coenzymes, via not having the pyruvate kinase active, via the starving fasting lipolysis and the acetyl-CoA concentration inhibiting the pyruvate dehydrogenase, and including the insulin and calcium activating the pyruvate phosphatase to activate it. You also have, if you have citrate, citrate will block citrate synthase, and this is done through competitive inhibition. And you also have rat poison, which is a conatase, or a rat poison is called fluorocitrate, and that is also known as rat poison, and that blocks iconatase. And you also have the isocitrate dehydrogenase. What it does, it is blocked or inhibited by NADH and ATP, and it is activated by calcium and ADP. So as I go through this, I'll kind of explain a little bit more so we can remember a little bit better. But for now, let's just continue. So the succinyl-CoA and the NADH block the alpha-ketoglutarase, and then calcium activate it. All right. So one of the key things here is pretty much a lot of the end products, if you have an accumulation of it, it will block that enzyme. So citrate synthase, since it's making citrate, if you accumulate too much citrate, then it will block this enzyme because it's saying way too much, slow down. Really similar to isocitrate dehydrogenase, it is pretty much saying, like, since it's making NADH, if it's making too much, it's gonna block that. And this whole product thing, at the end product of it is ATP. If it's creating too much of that, it will also block that. In contrast, if it doesn't have enough ATP, if it's not generating enough, then there'll be a lot of ADP, and there'll be a lot of calcium saying, hey, muscles, I'm using it, I need more energy, and that will activate the isocitrate dehydrogenase. And succinyl-CoA is a product of alpha-ketoglutarate dehydrogenase, so that will negatively inhibit it if there is an accumulation of succinyl-CoA, and NADH is also a product of it, so it does the same thing. And same concept of calcium saying, hey, like, I need more energy, activate now. And another key thing about alpha-ketoglutarate dehydrogenase is it is actually pretty well regulated. This is like #1 regulated pyruvate dehydrogenase, but #2 is alpha-ketoglutarate dehydrogenase because it also needs the same coenzymes as pyruvate dehydrogenase. And those coenzymes, do you guys remember any of them? NAD,
TPP? Or no.
Thymine?
Yes, TPP, right? Thymine. And then you have your lipoic acid. And we're missing one more. CoA. I like organize it. I guess if you want to look at it, it's like all these like 3-letter ones and then lipoic. So FAD, NAD, CoA, TPP. Just like, oh shoot, you're getting too fat. You've got like liposuction and lipoic acid. Okay. So these coenzymes are required at pyruvate dehydrogenase and alpha-ketoglutarate. If those coenzymes are not present, then those 2 enzymes do not work. Does that make sense? And then something that she mentioned is this succinate dehydrogenase is also functioning in complex 2 of the ETA or ETC. And I'm pretty sure she'll go over that on Friday when she does ETC. Do you guys have any questions so far? Okay, so what I did was I made a Google Doc of the diseases.
Do you want me to stop?
Yeah, you can stop.